Okay, so now let's talk about how microbial populations actually grow. So if you remember, when we're talking about bacterial growth, we're not talking about cells getting bigger. We're talking about cells getting more numbers. We're getting more cells. There's a couple of different ways that bacteria can grow. One is binary fission. Binary fission is the most common way that bacteria grow when one parent cell divides into two equal daughter cells. There's also budding. Budding is what's going on in this picture when the parent cell makes a little protuberance and that protuberance grows into a daughter cell. So you have a little protuberance, here it's getting a little bit bigger, here it's split off. Budding is not even. You have one distinct larger cell and a distinct smaller cell. In binary fission, your cell will copy its DNA, grow a little bit longer, then it'll start to constrict in, its plasma membrane starts to slip in, its cell wall starts to push in, until eventually it forms a cross wall. So you have two divided cytoplasms, and then this cell wall will split and you have two separate cells. At the end of binary fission, you have two identical cells. So this does not produce a mother and a daughter cell. This produces two daughter cells that are exactly the same. The amount of time it takes for a cell to divide is called its generation time. So just like in people, your parents were one generation, you're the next. So how long it took for your parents to have kids. Then when you have kids, that's the next generation. In bacteria, every time that a bacteria splits in half is a generation time. That's a new generation. Different species will have different generation time. Also, it's going to vary with different environmental conditions. If it's at a refrigerator, it's not going to grow very fast. If it's in an incubator, it'll grow very nicely. For most bacteria, a generation time is 1 to 3 hours. Although I think the fastest bacteria is like 20 minutes for generation time. So in 20 minutes, it's divided. In 40 minutes, it's divided again. After an hour, it's divided again. Because generation time is doubling every time, you're going from 1 to 2 to 4 to 8 to 16, 32, 64, 128, 200, 500, 1,000, 2,000. We're not going a nice little adding. We're doubling. We are going to use logarithmic growth. That's because if you try to just draw the chart, suddenly you're just exploding. Like, how huge would your chart have to be to keep going after this? Instead, we use logarithmic growth. This means that, one, you can chart things at a normal size, and two, these first few generations, it looks like nothing's grown for 10 generations, but that's not true. It's growing every time. So we're using a logarithmic growth. If you're into math, if you like to geek out about math, sometimes I kind of like my numbers. You can see how many cells are growing after so many generation numbers. So 2 to the 0 will give you 1 after you start with one cell after no generations. After 5 generations, you have 2 to the 5 or 32. You have 32 cells. After 20 generations, you have 2 to the 20 or over a million cells. And if you want the log number, that's how you would graph it. Okay, could a complex organism like a beetle divide by binary fission? Could you have kids by binary fission? Why or why not? Okay, then let's talk about the phases of growth. So when you have taken your E. coli and you have spread it out on a plate and you throw it into the incubator, that bacteria does not automatically just bam, jump into growth. It's first going to go through a lag phase. In the lag phase, it's kind of the smells the coffee, waking up, stretching, brushing its teeth, getting ready to grow kind of period. There's not a lot of cell division going on there, but the cell is getting ready to grow. It's making ribosomes. It's producing cell membrane. It's copying its DNA. It's doing the things to get ready to grow. The lag phase can last an hour. It could last several days. Then you have the log phase. In the log phase, that cell is prepared and now it is growing and growing and growing and growing as fast as it can grow. It is exponentially growing. Then the log phase is eventually going to be replaced by the stationary phase. In the stationary phase, now you've made so many cells that you're starting to get some cell waste products building up that kind of inhibits cells from growing. You're starting to use up a little bit of the food. There's enough food that some cells are still growing, but there's enough waste products that some cells are starting to die. So you kind of have this equilibrium where the number of cells that are growing is going to balance the number of cells that are being killed. Then eventually your cells will use up all of the food that's available there. There won't be any energy left for the cells to grow. There's so many toxins or waste products built up that the cells are now dying. You see a decrease in the number of cells. If you were to chart out the number of cells, then first you have the lag phase. The cells are not growing yet, but they're preparing to grow. So you still have just a nice even number of cells. Then the logarithmic growth, where there's plenty of cell growth happening. There's lots of good food. There's nice and good temperatures. It's just growing nicely. The pH is just right. Then the stationary phase. Maybe you've built up some acid. It's starting to get a little bit too acidic. You've started to eat out the food. There's not much left for everything to grow. Some things can grow, some things can't. So you have a stationary phase as the death and the production of cells are balancing each other out. Then the death phase, there's too much waste. There's not any food. The cells that are there are going to die. If you were to throw some cells on a plate, stick it in the incubator, and count the number of cells after every hour or day or whatever, you're going to see this exact phase happen. What would you see, though, if you had two mice? If you have a little mommy mouse and a little daddy mouse, and they love each other, and they get all romantic with each other, but they're in a fixed enclosure with a fixed supply of food. Do you have that same type of population curve? Would you still see mommy and daddy mice start to figure out how to have babies? Then they start having babies and grandbabies and great-grandbabies. Then some mice dying, some mice still being born. Then the food's all gone and everyone dies. Would you have that same kind of phase if we're talking about mice and a fixed enclosure with a fixed supply of food? Okay, we're going to finish chapter 6 in our next video, talking about how we can measure how much bacterial growth we have. I will see you back in our next video. Bye.